El nombre de mi presentación es el Evidencia Experimental y Natural de la plasticidad fenotípica a la humedad de suelo en condiciones invasivas de Cactoblasis cactorum. Entonces, las invasiones biológicas, más o menos, según los últimos cálculos en el 2017, representan cerca de 2.000 millones de dólares, lo cual casi que es el presupuesto nacional de muchos países, exceptuando Estados Unidos y China. Normalmente la manera en que controlamos estas invasiones es a través de esos análisis de riesgos de plagas, y hay un componente que es clave, que es qué tan riesgoso es que una plaga invasiva se mueva, ¿no? Regularmente esto se hace a través de similitudes climatológicas, como pueden ver en este mapa de la clasificación de Köppen, en donde tenemos similitudes entre diferentes continentes. Y solo para ponerles un caso, que es la polilla que vamos a estar hablando, pues en teoría esta polilla, pues ustedes pueden ver aquí todos los estadios, ya se ha movido por todas estas partes del mundo, abarcando pues diferentes condiciones climatológicas, algunas similares, otras diferentes. Y quiero hacer énfasis, por ejemplo, en esta, que es bien particular. Esta polilla es sudamericana, de la parte sur, y ya ha invadido zonas del Caribe que tienen unas condiciones tropicales, lo que quiere decir temperaturas, periodos de temperatura y precipitaciones distintos, y probablemente también humedades. Esta es la distribución nativa de Cactoblasis cactorum. Todos son registros que ya hemos georreferenciado. Si ustedes pueden ver que solo en los límites de la distribución norte hay registros en zonas tropicales. Estos son algunos de los mapas de nicho ecológico utilizando toda la distribución nativa y utilizando solo la región de donde se sabe o se conoce que se movió la plaga, y ustedes pueden ver resultados diferentes. ¿Qué quiere decir esto? Si solo consideramos la información de donde salen las plagas, donde conocemos, pues si hay procesos adaptativos o no a condiciones climatológicas, las predicciones pueden ser distintas. Y otra cosa importante es que de todas maneras las plagas invasivas o organismos invasivos, no necesariamente plagas, esta es una plaga, pueden presentar procesos de evolución rápida, adaptativos a las condiciones, o pueden presentar ya en su región nativa condiciones fisiológicas o ecológicas o comportamentales que la pueden preadaptar a esto. Como les contaba, esta es la región nativa, y ya hay estudios con diferentes marcadores moleculares que demuestran estructuras poblacionales genéticas distintas con COI, con marcadores de microsatélites y con SNPs. Y lo que yo quiero hacer énfasis es en estos dos grupos grandes en el centro de la distribución geográfica, en donde tenemos un grupo que está orientado hacia las zonas áridas de Argentina y otro hacia la zona subtropical, que sería esta clasificación de Köppen, que lo voy a seguir utilizando de ahora en adelante. Este insecto tiene varios estadios. La larva ingresa a los cladodios o tallos modificados de los cactus, sale de su hospedero, hace su pupa. Ese estadio se desarrolla en el suelo, en una capa de 5 centímetros los hemos encontrado, y finalmente salen las hembras y los machos que tienen este dimorfismo. Se ha demostrado de que el estadio pupal presenta diferenciaciones en características relacionadas con la aptitud biológica, como la temperatura. Sin embargo, la humedad, no se ha comprobado hasta el momento si la humedad de las condiciones del suelo donde ella se desarrolla afecta. En otros grupos en Cránvido, especialmente estudios realizados en China, han demostrado de que la humedad, el porcentaje de humedad puede tener un efecto sobre la supervivencia de Lepidópteros. En ese sentido, lo que nosotros hipotetizamos es que la disponibilidad de recursos de agua en los suelos donde la pupa está presente puede hacer que hayan procesos fisiológicos o de desarrollo que afecten a la pupa, o puede indirectamente fomentar la aparición de microbios o patógenos, y con esto afectar las características de rasgos biológicos. De esa manera, como que lo que nos planteamos es determinar en la condición nativa y con otros datos que recopilamos a nivel mundial, si efectivamente esta posibilidad de que Cactoblastis esté, por ejemplo, en la zona Caribe, que es subtropical, es producto probable de cambios evolutivos rápidos o preadaptaciones. Por lo tanto, el objetivo primero que nos quisimos poner a prueba fue caracterizar las condiciones de humedad de suelo en todas las regiones donde se ha reportado Cactoblastis cactorum. Como es una plaga que se ha utilizado como control biológico, pero luego se volvió plaga, tenemos muchos datos geográficos e históricos que nos pueden permitir saber si la especie se estableció o no en una región geográfica y queremos comparar esas condiciones. Y ya luego experimentalmente hicimos un estudio en el área nativa en donde tratamos de ver si había adaptación local a condiciones extremas de la humedad. Para esto lo primero que hicimos fue una caracterización de la humedad de suelo, utilizando publicaciones que tuvieran ya georreferenciadas las coordenadas o utilizando la descripción de las localidades y haciendo un proceso de georreferenciación siguiendo diferentes fuentes bibliográficas. Con esta información utilizamos estos datos satelitales de la NASA, que son SMAP, que son radar, que son ondas que envían los satélites, y a través de Google Earth Engine, en la capa superficial de 5 centímetros, caracterizamos la cantidad de agua que tiene esta capa superficial. Luego ya para el experimento, y luego lo comparamos con modelos generalizados, clasificando por origen geográfico, nativo, invasivo, australiano, sudafricano y caribeño y Estados Unidos, que es básicamente la región del Norteamérica. Luego ya para el experimento de trasplante recíproco hicimos colecciones de los insectos y del suelo. También hicimos la crianza de estos, una estandarización experimental que les voy a contar a continuación, porque es este método para comparar el fitness en condiciones de suelo nunca se había desarrollado. Hicimos una mezcla de varios procesos y ya finalmente hicimos la experimentación. Utilizamos estas dos poblaciones que son como la zona central de la distribución que les contaba y nos basamos en casi que en el centro de la distribución geográfica de estas poblaciones, que es la región de Entre Ríos, que ahora en adelante la llamaremos subtropical, y la región de Santiago del Estero, la provincia de Santiago del Estero en Argentina, que sería la región semiárida. Entonces, entonces aquí ustedes pueden ver los puntos geográficos donde trabajamos. Como ven, aquí en Santiago del Estero, que nos quedaba cerca a la región donde hicimos las experimentaciones, en muy pocos puntos logramos obtener las larvas, pero en la región nos tocó extendernos en toda esta región porque la disponibilidad de material biológico era bajo, dado que teníamos tasas de parasitoidismo muy alto. Entonces tocó extender el muestreo a toda esta región. Aquí ustedes pueden ver en cruz roja los sitios donde recolectamos suelos. Aquí ustedes pueden ver las condiciones de laboratorio en donde llevamos a cabo la cría de los insectos y estandarizamos muy cuidadosamente el punto cero, el punto de inicio de la experimentación, estudiando detalles del comportamiento de las larvas antes de empupar para que tuviéramos un tiempo cero efectivo. Aquí ustedes pueden ver la distribución de las recolecciones de muestras que hicimos, 189, ahí no alcanzo a ver, y 184 para la región de Santiago del Estero. Luego, para la estandarización de las muestras de suelo, como les decía, caracterizamos de nuevo en estos 3 sitios donde teníamos disponibles suelos, las condiciones de humedad utilizando este satélite, para probar efectivamente si había diferenciación en la disponibilidad de agua de esas condiciones, y hicimos una caracterización en laboratorio del punto de marchitez permanente que ustedes pueden ver acá. Ay, Dios. Que es la disponibilidad es el agua realmente disponible para procesos biológicos, porque el suelo es una esponja que absorbe la humedad y solo algún porcentaje de esa humedad está disponible. Para fortuna de nosotros, pudimos observar que en las dos regiones, una de las localidades presentan puntos de marchitez muy similares y eso nos permitió quitar eso como un factor adicional que nos pudiera estar afectando. Estas diferencias son resultados de procesos agrícolas que hacen modificación de los suelos propios de esta región. Luego, entonces, con esta caracterización utilizando el satélite, utilizamos fórmulas desarrolladas en trabajos de suelos como Brouwer y calculamos el porcentaje de humedad que debe tener cada uno de los suelos y ustedes pueden ver los contrastes de las dos regiones. Luego hicimos una caracterización del punto de saturación del suelo, qué tanta agua el suelo utiliza, utilizando un hobo para efectivamente demostrar de que el suelo estaba seco, el cual se secó con calor, y hacer un cálculo de la cantidad de agua que teníamos que adicionar. Aquí ustedes pueden ver una caracterización que realizamos y que también es un aporte que consideramos bueno porque utilizando estas condiciones de suelo de incubación de las muestras, caracterizamos qué tanto el tratamiento a lo largo del tiempo se mantiene para determinar así qué tantas veces hacíamos el recambio de suelo para que el tratamiento fuera homogéneo a lo largo de las, ay Dios, el tiempo. Luego introducimos, estas son las unidades experimentales, ponemos las pupas, mezclamos el suelo con la humedad correspondiente, medíamos el largo del ala. Estas son las medidas que tomamos: emergencia, número de días, longitud del ala. Estas son las variables predictoras: grupos genéticos, semiárido y subtropical, el trasplante, lo que recreamos, la adaptación local vista como una interacción entre ambas variables, el sexo, covariables que también medimos, que estaban relacionadas todas con la primera de ellas. Y como resultados, a nivel de la superficie del suelo, aquí ustedes pueden ver la caracterización de 495 puntos geográficos a lo largo del mundo, y ustedes pueden ver que la amplitud de suelos donde se desarrollan las pumpas es muy amplio y teniendo una diferenciación bastante marcada en suelos africanos y suelos de Norteamérica. El resultado de trasplante recíproco no nos arrojó una diferenciación entre las poblaciones de acuerdo a las condiciones de suelo que hay para la variable de emergencia o salida de las pupas, y para los días de desarrollo tampoco evidenciamos un patrón que nos indicara adaptación local. Si hay un efecto independiente de la población, el tratamiento semiárido tiende a retrasar el desarrollo, al igual que hay una diferenciación en el semiárido de salir a adultos más grandes. Para finalizar, lo que nosotros consideramos es que la preadaptación es como la característica que ha permitido que esta especie, la plasticidad fenotípica que tiene esta especie, ha permitido colonizar diferentes condiciones de suelo diferentes. Y algo que queremos hacer mucho énfasis cuando hagamos la publicación es lo que significa el cocón como una estructura que se ha demostrado que tiene condiciones de aislar la humedad, siendo una estructura pues muy importante para eso. Y algo interesante es que ustedes ven acá todos los grupos taxonómicos dentro de los insectos que tienen seda, y en estos grupos que están marcados en rojo son aquellos que tienen cocones. Lo que está indicando esto es que puede ser una estrategia que ayuda a aislar a la pupa de estas condiciones ambientales. A nivel práctico, lo que consideramos es que la plasticidad fenotípica nos puede estar mostrando una mayor capacidad de colonizar nuevos ambientes. Y en vista de las producciones, esa es la distribución de producción de Opuntia ficus indica, que es el nopal que se utiliza principalmente. Y pues todas estas son regiones marcadas en roja donde la larva todavía no ha ingresado. Tenemos zonas muy áridas como la norte de África, Chile, zonas tropicales como Brasil, lo que demuestra es que todas estas zonas están en riesgo. Y algo interesante es aquí está caracterizado los cultivos de nopal en México en cuanto a las condiciones de humedad, y ustedes pueden ver que el rango de datos disponibles según el Ministerio de Agricultura mexicano está en el rango de tolerancia de este insecto. En agradecimientos a los programas PAPIT y Postdoc de la UNAM que financiaron el proyecto, a estos colaboradores de Argentina que ayudaron en la fase de campo y laboratorio, a los agricultores y personales técnicos, y pues les agradezco mucho.
Well, thank you very much, Eimer. We will actually need to go on to the next speaker. So if you could stop sharing your screen. Just because the next speaker is going to start in less than a minute.
Okay.
All right, so go ahead, Jane.
Can you see?
We can see.
Can
And
you
I
hear?
will,
Sounds like you can hear. That's good.
yep, and I'm going to tell you to go now.
Okay, well, thanks for joining, everyone. You all know that natural habitats are increasingly restricted and fragmented, and that means that ambitions at the interfaces of population, evolutionary, and conservation biology need to understand what are the net genetic effects of dispersal and resulting immigration among the remaining habitat patches, and what are the ultimate implications of that immigration for the probabilities of local population persistence or extinction. And here we already know that immigration could have multiple genetic effects on individual and population fitness, acting within and across generations. Immigration could have multiple positive effects, for example, by alleviating local inbreeding and resulting expression of inbreeding depression. It could generate strong positive heterosis and offspring of immigrant native crosses, and it could increase local additive genetic variation and resulting potential for rapid evolutionary rescue. At the same time, we also know that immigration could have substantial negative effects by disrupting local adaptation and causing migration load, and also by causing outbreeding depression, especially following recombination in the F2s and subsequent generations. And we know that all of these effects can be substantial in theory and in practice, as evidenced by numerous experimental studies. But the net effects of natural immigration occurring within fragmented landscapes are much less clear. We need to know what are the net effects on individual fitness, on the emerging degree of effective gene flow, and ultimately on the dynamics and persistence of small remnant populations. And to answer these questions, we need to track the fates of natural immigrants and their descendants over several generations. And to do that, we can visit a small population of song sparrows that lives on Mandati Island in British Columbia in Canada. So here's a photo of Mandati in all its glory. It's a small population that typically holds between 20 and 45 breeding pairs of sparrows. And thanks to intensive fieldwork since 1975, we've compiled complete life history and pedigree data for the population, allowing advanced evolutionary quantitative genetic analyses. And one important point here is that although Mandati is an island, it's not particularly isolated or unusual. It's surrounded by lots of other islands and mainland habitat patches, many of which also hold song sparrows and some of which are very close by. So that means that Mandati receives occasional natural immigrants, roughly one a year on average, which can be of either sex. It's therefore very well suited to quantifying the immediate and longer term impacts of occasional natural immigration. And so far, we've identified 4 major genetic effects of immigration that I'll briefly summarize. First, analyses of extensive microsatellite data show that the immigrants are effectively unrelated to each other and to the local population at the time of arrival. That means that they do import new genetic variants and prevent the local degree of inbreeding from increasing, and that consequently prevents the expression of strong inbreeding depression in fitness, which we know is manifested in this system. So this implies that the immigrants are causing some degree of so-called genetic rescue of the recipient population. Second, we also know that the immigrants cause strong positive heterosis in the F1 offspring of the immigrant native matings, but then cause apparently strong outbreeding depression in the F2 grand offspring. And this is illustrated here for the fitness component of egg or embryo survival to a few days after hatching. And what you can see is that the F1 eggs have much higher hatching success than the natives, while the F2 eggs do very badly. And these same patterns also hold for other fitness components. And this apparent outbreeding depression in the F2s is especially notable. It's consistent with epistatic breakdown following recombination, and is really surprisingly strong for this kind of highly internet-connected metapopulation system. It implies that there may be some degree of local adaptation occurring within the range of regular natural dispersal. And following from that, the third thing we know is that advanced quantitative genetic analyses show that these immigrants are also importing additive genetic effects that decrease local juvenile survival. So they're effectively importing alleles with deleterious local effects. And this results in a strong migration selection balance, where the negative effects of incoming gene flow are counteracting local adaptive microevolution of juvenile survival. So these immigrants are effectively blocking the progress of local adaptive evolution and imposing a so-called migration load. Fourth, the other thing we know that the immigrants are doing is importing additive genetic effects that are altering the mating system. They increase the degree of genetic monogamy by slightly decreasing the degree of extra-pair reproduction. That in turn slightly alters the variance in male reproductive success across the population, which slightly alters the effective population size and hence the expected rate of reaccumulation of inbreeding. So, to summarize, these natural immigrants are having multiple, quite complex genetic effects acting within and across generations. They are introducing new local genetic variation, they're decreasing the degree of inbreeding and resulting expression of inbreeding depression, they're causing strong heterosis in the F1 offspring, but then outbreeding depression in the F2 grand offspring. They're introducing negative additive genetic effects that block the progress of local adaptive evolution, and they're altering the mating system and the resulting effective population size. So that's quite an impressive list of sins for a few incoming sparrows. The questions then are, what are the net impacts of all of these multifarious genetic effects on the effective degree of incoming gene flow and ultimately on the probability of local population persistence or extinction? To look at gene flow, we can use the full population pedigree to track an individual's expected genetic contribution to the population in each year following its arrival or recruitment. So this figure shows those expected contributions on the y-axis with years on the x-axis. It's females in red on the left and males in blue on the right. In both cases, the filled symbols denote recent immigrants and the open symbols are local natives that recruited to the population in the same year that the new immigrant arrived. And what you can see in females in red is that the female immigrants on average make much larger genetic contributions to the recipient population than contemporary natives, but that difference only really emerges 4 or 5 years after the immigrants originally arrived. And that's consistent with heterosis-enhanced introgression, where the high fitness of the immigrants' offspring, not so much the immigrants themselves, is spreading the immigrants' genes through the population. But you can see in blue that the same is not true for males. If anything, male immigrants make smaller genetic contributions than contemporary natives, and that's typically because the immigrant males have relatively poor mating and reproductive success. So overall, the net degree of gene flow into the focal population is greater on average and much more strongly female biased than we would have inferred by simply measuring the rate and sex ratio of incoming physical immigrants. So that's interesting. We apparently have heterosis-enhanced gene flow, but I also told you that these immigrants are importing additive genetic effects that decrease local juvenile survival and hence decrease the local population growth rate through that component. So that raises the question of what is the net genetic impact of immigration on the probability of local population persistence or extinction? And to answer that, we can use an individual-based stimulation model where we can vary the immigration rate and the magnitudes of inbreeding depression, local adaptation, and additive genetic variance in fitness, and we can quantify the probability of population extinction over many replicate runs given demographic and genetic stochasticity. And this heat map shows one summary set of results with baseline parameters set to broadly capture the song sparrow life history. The 3 main columns here illustrate immigration rates of 0, 1, and 2 new immigrants per year, shown in blue along the bottom. And the minor columns within those major columns are different increasing degrees of inbreeding depression. The 3 major rows are increasing levels of local adaptation from very weak to medium up the right-hand side in blue, and the minor rows within that are increasing magnitudes of additive genetic variance in fitness. And heat map colours show the probability of local population extinction, where dark red is almost certain extinction and light cream is almost certain persistence. And what you can see here is broadly intuitive. So with very weak local adaptation along the bottom row, increasing immigration into the focal population decreases extinction probability, especially given strong inbreeding depression. So that's genetic rescue. Meanwhile, with stronger local adaptation along the top row, increasing immigration from left to right actually increases extinction probability. So that's due to migration load. And in all cases, higher additive genetic variance, so that's increasing rows up within each of the major rows, decreases extinction probability because it's increasing the probability of rapid evolutionary rescue. Now, the black squares in the top row highlight the parameter space where we think the song sparrow population lies. And you can see that the system is really on a genetic knife edge where the positive and negative genetic effects of immigration are almost balanced. If anything, an increase in immigration from the left of those squares to the right-hand one slightly increases the probability that the recipient population will go extinct over an approximately 50-year time frame. And that's quite counterintuitive in terms of the generally expected effect of natural immigration into a small population in the context of a fragmented landscape. It implies that we should perhaps be quite careful and not assume that genetic rescue will be an inevitable outcome in that kind of situation. So, with this quick summary, I hope I've shown you how we can now estimate some quite complex quantitative genetic effects of natural immigration occurring in nature, and accordingly, how we can work towards a better and more predictive understanding of joint population and evolutionary dynamics emerging in fragmented systems. Thank you very much, and I'll be happy to take any questions.
All right, so we have plenty of time for a couple of questions. If you want to ask a question, use the raise hand function or type it in the Q&A. If no one comes up with any questions, then I will subject our speaker to questions. All right, so we have a question from Brian. Go ahead, you should be able to unmute yourself now.
Hi, thanks so much. That was such a wonderful talk. I was curious as to, can you say a little bit more about the way that you calculated migration load and how you predict the sort of net effect of that, like quantitatively?
Yeah, good question. So we did those analyses using the long-term pedigree data that we have for the population, where we can fit so-called genetic groups models, which basically partitions the change in total breeding value in the population into evolution from the original pedigree founders versus that coming from the new immigrants that arrived since the start of the study. And if we do that, what we can see is, as expected, an evolutionary increase in breeding value for local survival compared to the founder population. And that's not surprising because we know that there's additive genetic variance in survival, and of course there's strong directional selection on survival, so we expect a local evolutionary change. But at the same time, we can see that the genetic values imported by the immigrants are quite strongly negative compared to the founder baseline. So when you add those two things up, you effectively get a net change of 0, but it's because the local adaptive evolution from the founder population is being completely wiped out by the negative incoming effects from the immigrants. And so if you want to see more about those methods, there's a few papers now that are using these so-called genetic group methods in animal models to partition out the founder and immigrant effects.
So I will ask a question, actually. So I don't know if these are a species of particular conservation concern, but if these were being managed, would you have any suggestions based on your model results for the managers?
Yeah, it's a good question. So song sparrow is a very common bird across North America, as many of you will know better than me, but actually they're decreasing also a lot, as many things are, and habitats are fragmenting. So while there's no immediate conservation concern, I think there are some lessons, which is that we should just be careful about assuming that connecting populations is always going to be a beneficial thing, because it may be in some of these spatially heterogeneous systems that is not going to be the case. So I think that would be the take home.
All right. So with that, we're actually going to have to move on to our next speaker. So Inon, you got your screen shared. And we're going to wait just until it hits 11:30 to actually start.
Okay, just let me know.
Will do. This way we get it synchronized across rooms. People may be switching which sessions they're going to. All right, you can go ahead and begin.
Okay, so it's nice to be here with you. My name is Inon Sharf and I work at Tel Aviv University. I'm going to talk about ambush predators and whether they are really inferior to active predators, as it is continuously suggested in the literature. In the two photos below, we can see two related congeneric species of skinks. The one to the left is a whitey foraging predator. It chases the prey. The one to the right is a sit and wait predator. It doesn't move a lot. It just chooses a site and ambushes there. But a good point to start, perhaps a better point to start, would be to say a few words on foraging modes. So it's a continuum bounded by the two extremes, active and ambush predators. As I just said, the active predators chase after the prey, and most of the predators do so. A minority of predators just choose a proper or a suitable ambush location and wait for the prey to reach them. It is believed that active predators encounter more prey, and therefore they are more selective. They can choose not to consume everything they encounter, and they clearly pay some costs, the energetic cost of movement, and they are typically more exposed to predation. In contrast, ambush predators are supposed to have lower encounter rates with prey. Therefore, they are opportunistic. They cannot be very picky. They just eat what they get. And they save the energetic cost of movement. And because they are usually more camouflaged and they don't move so much, they are more protected against their own predators. It is now known that there are syndromes of foraging modes, a correlation of behavioral, physiological, morphology, and not only life history traits that go together with each foraging mode. The pioneering paper was that of Yuri and Bianca in '81. They worked or are working on lizards. And this is the set of characters they propose for both foraging modes. And I would like to focus on this. And as you can see, they suggested that the volume of prey captured for a unit of time for the sit and wait predators is lower than that of the active predators. The main goal of my talk is to ask, is it really that so? And if it is not always under which, if it is not always so, under which conditions do ambush predators do as well as active predators? So my goal was to examine the competition between active and ambush predators, and for this sake, I used an individual-based model. Individual-based models are in general a good tool for this purpose because they have a spatial component, which is difficult to implement using regular analytical models. And previous simulations that others people did, and also I did several ones before, so they showed an effect for the movement speed of predators and prey. For example, if the active predator is relatively slow, then the encounter rates are dictated more by the prey and less by the predator, and then maybe it's better to be a sit and wait or an ambush predator. It is also affected by the movement directionality of predators and prey. If the active predator does not use directional movement, but rather some tortuous movement, then it is inferior to other ones moving more directionally. It clearly depends on the movement costs of active predators. So the costlier or the movement is, then it is less beneficial to be an active predator. And other models show that the variance in capture success is higher for ambush predators. So some do very well and others do not, while the variance for active predators is usually lower. And it clearly depends on the positions of ambush predators. So some positions are more profitable than others, and I'll come back to that soon. Now I would like to say a few words on area restricted search. So area restricted search is a type of movement according to which the forager is moving in straight lines or in directional movement prior to the encounter with a prey item. But then after the encounter, after it encounters the first prey item, they switch to more tortuous or more non-directional movement in order to stay in the same patch. And the assumption is that the prey is clustered, and by doing so, they can find more prey. A good example is this ladybug or ladybird beetle. They hunt aphids, and they don't see so well. When they encounter the first one, they use a more tortuous movement, as you can see here. This is a paper from '85. Above, you can see the type of movement, more directional one before an encounter with prey, and below after. And this is a classic model of area restricted search. I designed two individual-based spatially explicit model. By spatially explicit, I mean that it takes into account the spatial component. I used a system of 3 trophic levels. The plants were sedentary, the herbivorous prey was mobile, and the predators were either ambush in the first model or both active and ambush in the second model. So now I'm referring to the first model. It's a game among 3 trophic levels. It asks what is the preferred strategy for each player. And in the absence of predators, herbivores should use the area restricted search to search for clumped prey. And let's assume that these are the clumps of plants. This is the herbivore. When it encounters the first plant, it will use more tortuous movement until it depletes the patch, and then it will resume directional movement. But then ambush predators should realize that and should locate themselves inside the plant patches, as you can see in this scheme. And this will cause the herbivores to stop using area restricted search because they may lose a meal, but they keep being alive. And then the plants can become more clumped. The predators realize that there is no benefit in being located within patches, and they also go out. But then the herbivores resume using the area restricted search, and we get therefore, and the plants become less clumped, and the predator enters back the patches, and we get cycles. So the simulation points to cycling phases. Let's call it a phase. So cycles between 2 phases. Phase 1 is when the plants are less clustered, the herbivores use area restricted search, and the predators are out of the patches. And the second phase is when plants are more clustered, the herbivores do not use area restricted search, and the predators are in patches, and so on and so on. The second model is actually the goal of this talk. So my question here is, when do ambush predators succeed as well as active predators? And when I say succeed, I refer only to encounter rate. So I simplify the cost of movement. Of course, if we add the cost of movement, so the situation even gets worse. So again, it's a similar model. It's a game among 3 trophic levels. The plants are in green here, the herbivores are in blue, the ambush predators are in red, and the active predators are in light blue or cyan. The active predators almost always do better. So this supports the known literature, and the simple reason is that they cover more space per unit time. So they cover 2 cells, whereas the ambush predator covers only 1. However, several factors can greatly diminish the advantage of active predator. So I calculated delta success, which is the prey encountered by active predators minus that by ambush predators. And the higher this value is, the better the active predators do compared to ambush ones. And you can see that the delta success is greatly affected by ambush positions of predators. So when the predators ambush inside patches, they do much better than when they are ambushing outside patches. There is a difference of around 72%. Next, I allow the plants to regenerate. It means that the consumed plant revived after some point. This is not imaginary. Some herbivores do not consume the whole plant. Some prey, so let's talk about seeds, for example, in the desert. So the wind can uncover new seeds and so on. So some prey in some plants or seeds indeed regenerate. Combining regenerate, so when I allow for regeneration, this difference is almost doubled. Inside positions become much better than before. Inside positions become even better when I combine regeneration with attraction among prey. Attraction among prey means that when there is one prey discovered a plant, it attracts some other in its vicinity, and then they all consume the patches. This is also not imaginary, as we know that some birds, for example, follow others, like a scenario of producers and scroungers and so on. So this makes even the difference higher, and in this case, the inside positions of ambush predators do almost as well as active predators. The best success of ambush predators was obtained when active predators use non-directional movement and plants regenerate. This is the highest difference between inside and outside positions, and interestingly, as you can see, that the error bars present confidence intervals. And here there is basically no difference between the ambush and active predators when predators are ambushing inside patches. And this is a case, for example, when ambush predators can do as well as active predators. And this is also an example for an interaction because non-directional movement has little effect on its own right, but together with regeneration, it does have an effect. So it is not enough to know how each characteristic of the system behaves in isolation, because together we can have some other effect or different effect. Talking about interactions, so I would like to present a few other examples of interactions among tested variables or characteristics of the system. For example, uncertain capture, when the predator does not always capture the prey, which is, I think it's a realistic scenario. So when uncertain capture is allowed, then the differences between inside and outside positions are blurred, as you can see here. But if we combine uncertain capture with attraction of prey to each other, this rescues in some way the positions inside, and we get a significant difference. And attraction by itself has little effect, so this is another example of an interaction between two variables. Giving up on switching to area restricted search had little effect. By giving up on switching to area restricted search, I mean that when plants drop in density, when they are all exhausted, so the herbivores stop using area restricted search and they just move in straight lines. But combining it with regeneration greatly decreased the advantage that regeneration provided in the absence of giving up on area restricted search. And this is another example of an interaction. So to conclude, ambush predation is inferior in its encounter rate. So I think what was expected, I support what is expected. But more interestingly, we cannot know inferior to what extent. It's hard to know it without knowing many properties of the system. For example, where are the ambush positions? Is food consumed in total, or does it regenerate? Do prey attract each other? What are the movement characteristics of the prey or the active predators? And even if we know all that, so some interactions can produce unexpected outcomes. So we have to really know well the system in order to make predictions about the relative success of ambush and active predators. And ambush predators may have similar encounter rates to active ones if they ambush inside plant clusters and if their herbivore prey use area restricted search. And I would just like to finish with highlighting the importance of individual base models and their spatial aspects, because I don't know how to do it without a simulation using an analytical model. So thanks a lot for listening, and if we have time, I'd be happy to answer questions.
Yes, we should have time for at least one question. Well, if no one wants to ask it, I will ask a question. So in terms of your simulation, have you thought about adding an individual-based model of what it is the predators are paying in terms of the cost? Like, would there be a second order predator you put in there or some environmental feature?
Right, this is a good point, and it's clearly possible. I think if I add the cost of movement for predators, so the ambush predators will certainly do better. I think it will be even more interesting to have different costs for movement. For example, I think that going in straight lines will be cheaper, but going in more tortuous movement, using more tortuous movement, should be more expensive. There are even several papers on humans that if we turn a lot, so we burn more calories. So we can also implement that and test how it affects the usage of area restricted search, for example. So this is a really good point. I should do that, I guess.
All right. We are now actually going to switch on to our next speaker. So Eldrin, do you want to share your screen? And then we're going to wait just until it hits 11:45, which we're close to, but not quite there yet. Another 5 seconds or so. All right, you can get going.
Yes. Okay, so I don't know what time is it, but in here it's morning, 12:30 in the morning. So good morning to everyone. I'm going to present this study about Simbella janishi bloom and the application of DNA metabarcoding to provide insight on the microbial diversity, bloom formation, and potential threat. Okay. Oh. Okay. So Simbella janishi is actually an invasive benthic diatom species which causes adolescence blooms in rivers. Basically, this diatom form thick and dense mat, which is very similar to rock's knot, which is formed by Didymospina geminata. And as of the moment, it is being considered as a major threat for Japan's river ecosystem. So to date, only limited information is available on the possible causes of Simbella janishi bloom in rivers here in Japan. But it is suggested that roles of different microorganisms such as diatom, bacteria, and fungi can actually give clue to the reason why these benthic diatom species are actually growing in rivers in massive amounts. Thus, the objective of this study is to assess the effects of Simbella janishi blooms on diversity, assembly, and potential functions of microbial community in rivers, and to identify key microbial species that are associated with the Simbella janishi bloom using DNA metabarcoding, and to explore the potential growth-promoting activities in vitro of bacterial communities and associated metabolites on clonal cultures of this diatom. So basically, we did sampling from 7 rivers in Japan wherein evident bloom of Simbella janishi is present. We collected 2 types of samples. So the first one is a benthic sample labeled as benthic negative janice. So this is actually rock surfaces without evident growth of Simbella janishi. And the sequence reads of this sample is less than or equal 1% of for the Simbella janishi. So the other one is actually a stone or rock wherein evident growth of Simbella janishi is present, and we called it as matte positive jani. So these are actually samples with sequence reads that are greater than 1% of Simbella janishi. So these samples were, we did DNA extraction and PCR, and we did metabarcoding analysis using specific primers specific for diatom, bacteria, and fungi metabarcoding, and we did this analysis, the alpha and beta diversity analysis, Lepsi analysis, and concurrence network analysis, and we actually also predict the metagenomic prediction of bacterial functions using PyCrust2. And the result of this PyCrust2 analysis was further validated by doing growth experiments with clonal cultures of Simbella janishi, and then growth experiment using zeatin and co-culture experiments in some bacterial isolates from this associated with this Simbella janishi. So for the results, so we found out that the bloom of Simbella janishi is actually associated with a decrease in the bacteria bacteria and diatom species richness and evenness as supported by the amplicon sequence variance counts, the different alpha diversity indices, but not in fungi. So this is actually the graphs showing the math. This is the math, and then this is the benthic samples. There's actually the significant decrease for bacteria and diatom, but not in fungi. We also identified 71 microbial taxa using Lepsi analysis that are differentially abundant in Simbella janishi mat. And there are a lot of different microorganisms for diatoms. There are 6. For bacteria, there's so many, and then for fungi, it's only 1. And these microbial taxa are actually associated to have biological functions such as phytohormone producers, and as well as nutrient producers, which actually support the growth of this, which may actually support the growth of this Simbella janishi during its bloom in the river, in rivers. So we also did co-occurrence network analysis to understand the relationship of the different microorganisms associated with the bloom of the Simbella janichi mat. So we found out that two organisms, the Flavobacterium, which is a bacteria, and Anicia paliacea, a diatom, is actually have direct or positive interaction with Simbella janichi, showing, which means that this organism shows cooperative or synergistic relationship. So on the other hand, Melosaria variants, a diatom, and also Celepra nigri, also a diatom, tend to have a negative interaction against Simbella janishi, showing that these two diatoms are actually have a competitive or antagonistic relationship. So based from this co-occurrence network analysis, we also identify co-occurring and differential abundant species in Simbella janishi match, which include the Flavobacterium, Nichia paliacea, and Celapora nigri. So these um, um, microorganisms are not just differential abundant based on Lefse analysis, but also they are co-occurring together with Simbella janishi, which actually suggests that these microorganisms could play an important role in the bloom formation of these diatom. So the PICRAS analysis, we actually find out 40 keg or 12 pathways that are overrepresented in Simbella janichi mats, and these pathways are associated with carbohydrates, nucleotide, and amino acids, as well as vitamins and cytokinin synthesis. And one particular pathway that we are interested with is this zeatin biotin biosynthesis pathway, which is overrepresented in mat samples, or in the Simbella janichi mat sample. So what are zeatin? So zeatin are a class of phytohormones belong to cytokinin, and basically these hormones being produced by bacteria to help the host organism by enhancing growth via promoting cell division and nutrient uptake. So the question is it possible that this zeatin can positively influence the growth and proliferation of the Simbella janishi? So we did culture experiment. Basically, we grew Simbella janichi culture using WC medium at varying concentration of zeatin and grow them for 21 days and observe and did cell counting and measurement of the stalk length. So we found out that the cell number and stalk length of Simbella janichi significantly increased with higher concentration of zeatin, particularly the WC medium supplemented with 250 micromolar zeatin was considered as the best growth medium for this organism. So this is a fluorescent image showing the different setups, and we found and showing that that WC medium supplemented with 250 micromolar zeatin showed the best growth for the clonal cultures of Simbella janichi. We also did co-culture experiments with bacteria isolates. We isolate different bacteria associated with this Simbella janichi, and we actually use an axenic culture of Simbella janichi. This is actually an antibiotic treated Simbella janichi culture acclimatized in the laboratory, and we co-culture it with different bacteria. So these 4 bacteria that I listed in here are the 3 bacteria that I listed in here are the bacteria that was recovered in during isolation from the cultures of Simbella janichi samples that we collected in the field. So basically, they are the common bacteria that we recovered using our culture medium, and then we grow them, and we did cell counting, and then we compared the specific growth rate. So we first compared the xenic cultures. So this is a xenic cultures. This is the one with not antibiotic treated, and compare it with the growth of the axenic culture or without bacteria. And we found out that the axenic cultures of Simbella janichi cultures has lower growth rate as compared to the xenic cultures. But when we supplemented with Pseudomonas GB1, we actually show that there is actually it exhibited a characteristic growth which is comparative to the xenic cultures. So using this one. And when we actually co-cultivated with other two bacteria, which is the Gentilobacterium lividum and Eromonas pofafi, we found out that they actually cause a decrease, but the difference in terms of specific growth rate and maximum cell number is not significant as compared to the xenic cultures. So from this, let's focus on Pseudomonas. Pseudomonas are actually gram-negative bacteria, which are rod-shaped. They are known to have symbiotic relationship with several strains of microalgae. So basically, this group of microorganisms are actually known for acidophore and phytohormone producer, and they produce hormones such as zeaxanthin, auxin, and indole acetic acid. Also, these bacteria are biofilm polymers, which can actually help the diatom to promote interaction with other beneficial microorganisms, and also they degrade different organic matters, which actually can also help improve the nutrient uptake of the diatom. So here is actually just a bacterial community composition of a xenic culture of Simbella genici, and we found out that Pseudomonas is actually one of the dominant bacteria associated with the clonal cultures also. So from this, it may seem that these Pseudomonas strain may serve as a biomarker or indicator species for detection of Simbella genici blooms in high-risk liver. So there's still a lot of analysis that needs to be done to prove this, but um, it may be used in the future study with regards to this organism. Okay, so for the conclusion, co-occurrence between microbes and Simbella genici suggests that specific microbiota are associated with formation of Simbella genici bloom. The second one is the presence and colonization of Simbella genici in livers cause a reduction in diatom and bacterial diversity, but not in fungi. So zeaxanthin promotes cell proliferation and stalk formation in Simbella genici, and then Pseudomonas is capable of enhancing Simbella genici growth by exhibiting growth characteristics that are comparative to that delivered by the xenic cultures of Simbella genici. And with that, thank you very much.
All right, so we have time for a question or two if anyone wants to ask something. Go ahead and raise your hand. That's probably going to be faster than trying to type something into the Q&A. All right, if no one else is going to ask something, so what do you have planned next? Like, what's the follow-up to this most recent study?
So basically, what I'm doing right now is I do microbial dynamics. So I monitor it actually, the complete bloom of these diatoms during its bloom right now. So I do monthly sampling and then actually doing checking the dynamics of the microorganisms, changes of microorganisms during the start until the end of the bloom. So that's I can know which actually organism is actually increasing or decreasing, and at the same time really affecting the growth of this organism. Yeah, and also we plan to also do metatranscriptome analysis of this bloom. So also start, middle, and then end, just to compare the gene expression pattern of the microbial community associated with the bloom of this diatom. So yeah, that's it.
All right, we've got just 1 minute until our next speaker is going to go. Now for our next speaker, we have the recorded video, but we also have the speaker here in the room. So if I understand correctly, we have the video because there was some question about internet connectivity. So Rachel, do you want us to still play the video and have you just answering questions during it, or what do you want to do?
Yeah, let's do that to make sure that I don't drop off during the middle.
All right, so our volunteer here is going to share their screen and play the video, but for all of you who are in here in the session, you can start typing questions into the Q&A while the talk's going, and Rachel will be able to answer them as long as she still has an internet connection.
Perfect, thank you.
And then E, you can start when you are ready.
Thank you to the organizers and to all of you in the audience.